Okay, so
Yeah.
hello everyone. It is a pleasure to be here today to share about a topic that I feel is exciting, namely the recording of neural spikes from micro-ECOG or micro-electrocorticographic arrays and their potential application for brain-machine interface. So my name is Jack Low and I am a neurosurgery senior resident at the National Neuroscience Institute of Singapore. So for this talk, I will touch on the current state of the micro-ECOG technology from the broader literature, as well as present some of my own findings in this area. So I have no financial disclosures to make. Brain-computer or brain-machine interfaces can be divided into invasive and non-invasive systems with a trade-off between factors such as spatial temporal resolution, spatial coverage, and the degree of invasiveness. So on one end of the spectrum, non-invasive BCIs rely on external sensors like EEG that we have just heard about, and MEG and fMRI to detect neural activities. They typically have relatively low spatial temporal resolution, when compared to invasive implants. But however, non-invasive BCIs are advantageous as they do not require surgical procedures that will put the neural tissue at risk and are therefore safer, more accessible, and more scalable. Invasive BMIs, brain-machine interfaces, on the other hand, offer high spatial resolution information, which arguably enable fine-grained control. So among invasive interfaces, ECOG and intracortical microelectrode or MEAs are the most commonest types. And conventional ECOG arrays, as you can see on the top image, typically uses larger electrodes with sizes in the millimeter scale. And ECOG, these ECOG arrays sample from a broad area of the brain surface. And moreover, these electrodes are spaced relatively far apart with pitch ranging from a few millimeters to a few centimeters. And these systems are used routinely in clinical settings like epilepsy monitoring and tumor resection surgery, for example. But they can also be chronically implanted in some cases for BMI applications. And micro-ECOG, in contrast, employs smaller electrodes, typically on a scale of tens to hundreds of microns, resulting in higher electrode density. And this allows it to record from smaller populations of neurons. And the image below here actually show that two examples of micro-ECOG arrays by two separate groups with very small electrodes and dense arrangement. And also you can see the scale of such micro-ECOG arrays with respect to the brain in the image right at the bottom. So this is an image of a typical conventional 4x5 ECOG array taken intraoperatively, used in epilepsy surgery to record and identify seizure focus. So the contacts have an exposed diameter of 2.3 millimeters, and they're spaced roughly about 1 centimeters apart. So for such conventional ECOG grid, the 5 square millimeters or so of brain surface under each contact contains about 500,000 neurons. So quite a large number of neurons. And the activities of these neurons manifest as current dipoles in the vertical axis. And when these signals combine, they generate an ECOG potential that can be measured at the surface of the brain. And for detection of large amplitude, synchronous burst firing activities like seizures, large conventional ECOG arrays like this is sufficient for that purpose. And on the other hand, micro-ECOG arrays like this one shown here has a contact diameter of only 30 microns. And the diagram on the right shows the relative scale of a conventional ECOG array, as shown in the previous slide, as compared to a micro-ECOG. Going from 2.3 millimeters in contact diameter to 30 microns or 0.03 mm, there is approximately a 6,000-fold reduction in the surface coverage. And this significant decrease in the electrode surface area raises important questions about exactly what types of neural signals can be effectively recorded using such micro-scale electrodes. And once again, this diagram highlights the different scales at which neural activities can be recorded. So one key question is whether micro-ECOG electrodes can capture neural spikes or action potential. which are fundamental units of communications between neurons. And these spikes can either be single units or multi-unit activities, or SUA, MUA respectively. And single units in particular are electrical signals generated by single neurons and typically only recordable with invasive intracortical electrodes implanted directly into the brain tissue. As you need to be really close to a neuron in order to pick up its actual potential extracellularly. So why are spikes important? So recording neural spikes or action potentials from individual neurons is crucial because each neuron or a local cluster of synaptically connected neurons possesses unique characteristics in terms of firing rate, their firing pattern, and response profile to various stimuli. And this diversity within local neuronal populations highlights the importance of capturing fine-grained neural activity. And in regions like the motor cortex, even within a very small area of the brain, there exists a diverse population of neurons with distinct and different tuning characteristics. So for instance, here we see that each neuron on the left set of images, each neuron preferentially fires in a particular movement direction, as represented by the colors in these vector maps based on recordings from very small intracortical arrays implanted in the motor cortex and the premotor area. So the richness and diversity of neuron level information permits high degree of freedom decoding and fine-grained control of any vector devices. And in another example, in the right set of images, in the somatosensory cortex, deep and superficial cutaneous sensations are mapped to different individual channels on these MEAs. And despite covering just a few square millimeters of brain tissue, MEAs provide spatial precision in capturing sensory information. And these spatial precisions would be lost if signals were summated over a wider area, as in the conventional ECOG or EEG-based setups. So one key fundamental limitation in intracortical array is that they destroy brain tissue, and the insertion process itself is really quite traumatic to the brain. And many studies have shown that these shanks leave a sizable hole in the brain parenchyma, as you can see here. And this disruption triggers a cascade of reactive inflammatory responses from astrocytes and microglia aggregating around the implanted electrodes over time. And in the long run, these inflammatory reactions can actually lead to progressive neurodegeneration and signal loss. And there have been recent attempts to miniaturize these shanks and have non-immunogenic coating materials, for example. But as it stands, this still remains a major consideration for such invasive implantable arrays. And this is another diagram showing the typical time course of inflammatory changes. And initially, the electrodes sit in very close proximity to the neurons. So we can see as those blue cells on this image. But over the course of weeks, astrocytes and microglia form a shield around the electrode shanks and insulating the neurons from the electrodes. So intracortical arrays will therefore not be very scalable, at least with the current designs, as the risk of causing injury to the neural tissue and complications like bleeding and inflammatory reactions are cumulative and scale with the area of coverage, limiting how wide a cortical coverage we can achieve. So one question to consider is, without having to penetrate the cortex, is it actually possible to record single unit activities just from the cortical surface? In other words, how far can spike level signals travel to reach the recording electrodes on the brain surface? It's clearly not possible with the conventional ECOG systems to detect spikes, as the electrode sizes are simply too big. And as I mentioned, the activities are summated over a large population of neurons. But however, with the kind of microscopic electrodes used in micro-ECOG arrays, is it then possible to record spikes? And if yes, from which cortical layer do these spikes emanate from? So Chestek's group in Michigan has actually done a very nice simulation study looking precisely into this topic. So once the electrode sizes get small enough down to the micrometer scale, as in the case of micro-epochs, the population of neurons that lie in the outermost layer of the cortex, that is layer 1, those are the closest to the surface recording electrodes of the micro-epoch array. They become a very important population of neurons to understand and characterize. So when you have a large epoch array, these relatively small and sparsely distributed layer 1 neurons are practically invisible to the array, as the electrical dipoles and field potentials from the deep layers, which contain a lot more neurons with larger cell bodies, as you can see here, dominate the signals. So this group then simulated the kind of neural signals recordable with micro-epoch arrays, with small sub-millimeter electrodes resting directly on the peer surface, as you can see in the middle image here. So they found that signals originating from layer 2 and 3 and also layer 5 pyramidal neurons, which are considered the typical targets for intracortical recording. So those signals, they actually reach, those signals reaching this micro epoch array at the brain surface have very small signal amplitude on the order of 1 to 2 microvolts as per this simulation. And I'm referring to the image, the diagram on the left. And in reality, it's not discernible from the noise level. So on the contrary, neurons in layer 1, because they are so close to the electrodes lying on the surface, they can actually generate comparatively large signals with amplitudes on the order of 60 to 100 microvolts. So which will indeed show up on the micro-epoch array, and it is going to be significantly above the noise RMS in most cases. And also importantly, when the electrodes are appropriately spaced, an individual neuron, for example, this black neuron here, on the right image, will produce a strong potential in one electrode, but a very weak potential in the adjacent electrode, just 30 microns away. And the adjacent electrodes can, for example, be used as a reference, as they will receive the same local field potentials from the surrounding brain tissue, but only the spike activity from a neighboring layer 1 neuron will show up in any single channel. And the fact that layer 1 has low neuronal density, as per the previous slide, actually helps to keep these neural spikes separate between appropriately spaced electrodes, with little cross-contamination by neighboring layer 1 neurons. So as part of our own ongoing work, we have elaborated on this estimation by assuming an effective spherical radius of 60 microns from the electrode, where neural signals can be discernible from the noise floor. So assuming the same neuronal density as the numbers given by the study that I just mentioned, which is around just slightly under 10,000 neurons per square millimeter, per cubic millimeters, this comes out to approximately 3 neurons within this effective three-dimensional radius, which is actually a very manageable number of neurons to spike sort from the multi-unit signals obtained from the array. So at this point, we just want to move on to talk a bit more on the different layers of the cortex and their electro
Check, hello. Can anybody hear me? Please let me know in the chat. I think we lost Check for a moment. So let's see if he logs in again. Maybe his internet crashed. Any questions in the meantime about the group formation, maybe in the hackathon? Now it's time to ask. think Jack lost the connection because we can still hear each other. I'm waiting for him to log in again.
Yeah, I think I got disconnected.
Yes, we lost you for like 1 minute, Jack. So maybe
Okay,
you can just start with this slide again.
this slide again. Okay, yeah.
Thank you.
So, I mean, at this point, I just want to talk a little bit about the different layers of the cortex and also their electrical properties. As you can see here, we have two, understanding the kind of cortical layers and how neural signals are processed within these layers will actually be very important and relevant in understanding the nature of spike signals that we can record from micro-electrocorticographic arrays. So the cerebral cortex is organized based on cytoarchitecture into 6 distinct layers and their corresponding sublayers. And these layers are numbered from the outermost layer 1 to the innermost layer 6. And intracortical arrays primarily record from deeper layers, such as layer 5, with large pyramidal neurons that generate very strong action potentials. So in the motor cortex, some of these neurons give up axons which form the cortical spinal tract, for example. And this tract is involved in the control and execution of voluntary movements. And outermost layer 1 is fairly sparse in neurons, as I mentioned before. And their primary roles are thought to be involved in the modulation of cortical activities, integrating inputs from other brain regions, and potentially involved in neuroplasticity and the refinement of neural circuits. So there's also a prominent vertical flow of information between the different cortical layers. And the neurons, their axons and dendrites are vertically arranged in what pioneering neuroscientists like Mountcastle called vertical columns, as you can see on the schematic diagram on the right. So these vertical columns span multiple layers of the cortex, extending all the way from the pale surface all the way down to the white matter below. And due to their dense connectivity, the firing activities and the response profile of these neurons are often highly correlated. So this means that each column represents a functional unit of cortical processing, with neurons within the column sharing very similar response properties and participating in common computational tasks. So columns can vary in width across different cortical areas and species, but they are typically on the order of a few tens to a few hundred microns in diameter. And owing to this vertical flow of information, including to and from layer 1 neurons, the recorded activities of layer 1 neuron will mirror to some extent the response properties of deeper neurons in the same vertical column. So the columnar organization of the cortex has been described in various cortical areas, including sensory, the visual, and the motor areas. As you can see here on the left image, for the primary somatosensory cortex, or S1, cortical columns are organized somatotopically, with adjacent columns representing adjacent parts of the body. And once again, neurons from different layers in the same vertical column, they basically share the same circuit, and they share similar tuning properties and somatotopic representation. And in the primary visual cortex, V1, such as this study on tree shrews in the middle image, the cortical columns are organized into orientation columns, which respond selectively to visual stimuli of different orientations. And neighboring patches have similar orientation preferences, and they tessellate across the entire visual cortex. And lastly, the same has been observed in the motor cortex, with neurons within a motor cortical column sharing a very similar preference for the direction of movement. So beyond vertebral columns, these layer 1 neurons also play an additional role in an intracortical circuit, with horizontal or longitudinal projections emanating from layer 1 neurons that make contact with axons and dendrites of a wide variety of cells, both locally and in distant areas. So you can see on the myelo-architectural stains on the right image here that in layer 1, there are prominent horizontal striation patterns like the Cajal-Ratzius stripes, for example. So these intracortical circuits, they serve an important role in the sampling and integration of varied information across different cortical areas. So layer 1 neurons are actually quite poorly understood, and only recently are these neurons classified by various groups. And in this study, Schumann and colleagues characterized the largely GABAergic interneurons in layer 1 and classifying them into 4 types. So we have the neuroglial cells, the canopy cells, the alpha 7 cell, and the VIP cells, and each with a unique molecular profile, morphology, and electrophysiology. So unlike other layers, most layer 1 neurons appear to be unique to the layer, so highlighting the specialized character of signals processing that takes place in L1. So again, the same group actually gave a further summary of the different neuronal subtypes classified by their molecular profiles. So broadly speaking, the neuroglial cells and the canopy cells have exons confined to layer 1, and the alpha 7 and VIP cells have exons that descend down to the deeper layers. So these cells can be further divided electrophysiologically based on the spike characteristics. So for example, neuroglial cells are also known as late spiking cells in the rest of the literature due to the late timing of their spikes. And once again, layer 1 contains neurons both with dendritic and axonal projections confined within the layer 1 itself, as well as those that project down to the deeper layers. And in particular, the late spiking neurons, or LS neurons here, which morphologically correspond to the neuroglial cells as per the previous table, have inhibitory GABAergic synapses onto pyramidal neurons, as shown in this patch clamp experiment, where current injection into the presynaptic late spiking layer 1 neuron results in a hyperpolarization response in the postsynaptic pyramidal neurons. So on the other hand, pyramidal neurons have excitatory projections to these late spiking neurons, as shown here on the right image, that an action potential in the pyramidal neuron produces a polarization in the late spiking layer 1 neuron. So essentially, experiments like this show that layer 1 neurons are electrically coupled to deeper neurons, whose firing activities we are actually more familiar with in various BMI applications with the intracortical arrays. So another example of previous work looking at layer 1 neurons is this study of cat visual striate cortex by Martin et al. In 1989, where they actually mapped out the receptive fields of layer 1 neurons in response to light bars of different orientations presented at different parts of the visual field. So layer 1 neurons do have response selectivity, pretty much like the deeper neurons, as you can see in this example and many others. And also through histological studies, they mapped out the patterns of axonal and dendritic contacts between the layer 1 and pyramidal neurons in the deeper layers, demonstrating that they are frequently coupled through monosynapses. So therefore, layer 1 neurons inherit some of the response characteristics of the pyramidal neurons, again, the usual recording targets of intracortical arrays. So beyond local synapses, over longer distances, these layer 1 neurons also have been described through tracer studies and histological studies to be connected to the rest of the brain through, for example, the thalamocortical network, integrating information across a wide range of cortical areas through deep brain structures like the thalamus. So this diagram here basically shows quite extensive axonal and dendritic projections across to and from different regions, including different cortical regions, as well as deep brain structures. In this study by Kaneko and colleagues, they found that different parts of the ventral anterior and ventral lateral thalamus project to different layers of the motor cortex. And specifically, inhibitory thalamic neurons project to the layer 1 of the cortex, while excitatory neurons project to the lower layers. So there is some functional and connectivity kind of differences between the different layers. So these are some images taken from Schumann and colleagues, which show the connectivity patterns of various afferents to the primary somatosensory cortex, S1 in mice, demonstrated through fluorescence labeling. So each image here, each column, each image here shows the afferents coming in from the respective brain regions as per labeled below. So like areas like the secondary somatosensory cortex, S2, or the secondary motor cortex, M2, and so on, converging onto the S1 barofield cortex and mice. And it's quite remarkable, just looking at the concentration of, for instance, labels in layer 1, showing just how extensively connected layer 1 is to the rest of the brain. So in view of this rich and dense mesh of long-range projections providing signals to the tuft dendrites of the pyramidal cells, layer 1, again, is the most outermost layer that is the closest to the micro-electrocorticographic array, serves as a predominant input layers for top-down projections, top-down information, integrating across the different cortical layers. So this may in turn contribute to the phenomenon that information from elsewhere in the cortex, elsewhere in the brain, is represented in a distributed manner across different brain regions because of this neuronal connectivity. So for example, place cell-like activities, traditionally thought to be confined to the hippocampal regions, can be detected in areas like N1 and premotor cortex and S1. So in this experiment by Ain et al., the monkeys navigate a motorized wheelchair under BMI control from three possible starting points, as represented by the green circles, all the way to a grape dispensers, as shown by a cross on the left. And on reaching the docking zone, the monkey initiates a reaching movement to get the grape from the grape dispenser. So simultaneous recording with intracortical arrays found place cell activities across different sensory motor cortical regions, as shown on these heat maps of representative neurons. This are on the right. So traditionally, place cells have been, as I mentioned, associated with hippocampal regions. So this finding is interesting because it shows that spatial information can be distributed throughout the cortex, probably serving as context cues to guide local cortical processing. So there is also evidence that layer 1 neuron is a major participant in such distributed neural processing. So histologically, we have seen that layer 1 neurons have rich connections with a myriad of brain regions, both from the cortex as well as from deep structures like the thalamus or the entorhinal cortex. And in another study by Messick and colleagues here, layer 1 neurons in the visual cortex were found to carry a great deal of sensory and motor-related information, as quantified by 2-photon calcium imaging during a behavioral task. So in their experiments, head-fixed mice were habituated to freely move on a treadmill. So that when a mice's locomotion speed exceeds a certain threshold, they label the period as a run period, corresponding to the red bars here. So they basically they measured the layer 1 neurons at different depth between layer 1. And at specific intervals, a few different stimuli are presented to the mice. So we can have visual in the form of visual pattern, as you can see on figure A, auditory in the form of a noise burst, and tactile stimuli in the form of whisker touch. They basically found that despite being the visual cortex, only slightly under 50% of layer 1 neurons are sensitive to visual stimulus. And in fact, very slightly more neurons in layer 1 of the visual cortex are sensitive to the locomotion speed, which I think is interesting, and to auditory noise stimulus and whisking movement. So as you can see on this bar graph here, with the y-axis being the fraction of neurons responsive to each of the stimulus as measured by the calcium imaging. So again, this is interesting because this challenges the idea that the visual cortex is just for visual processing, when in fact a great deal of contextual information is also present in this area. and specifically in layer 1 neuron, with direct evidence showing this. And indeed, it has been proposed, well, I guess fairly recently as well, that layer 1 plays a central role in the integration of convergent information from different memory-related structures, such as the hippocampus and the amygdala, as well as from sensory areas of the cortex. So these input, in fact, in turn, guide local plasticity in the cortex. So recording from these layer 1 neurons, which lie the closest to micro-ECOG arrays, as I mentioned, may give rise to a few advantages as opposed to recording from deeper neurons. So firstly, because layer 1 is an important site for top-down integration of information with extensive cortical, and cortical-thalamic projections, as we have seen earlier, we may be able to extract some information features that are derived from cortical areas distant from the area that the recording is done on. So for example, locomotion speed and auditory information from the visual cortex, as per the previous example. And also another related point is that layer 1 is frequently the first point of contact, the landing zone for exons coming in from elsewhere, before the information is passed down to the deeper layers. And this means that we may be able to better study neuronal connectivity patterns between different brain areas in this way, especially if we scale up the micro-ECOG array, the span of the micro-ECOG array, to cover large brain areas while retaining the spatial resolution to get neuron-level electrical signals. So these conjectures, however, need to be further tested in future studies, and I guess it's a relatively new area. So going back to micro-ECOG arrays proper, so there is fairly consistent evidence, as I've shown so far, that there are actually many things going on within layer 1. So both in terms of cortical processing along the same vertical column, with very similar response characteristics in the cells in this column, as well as conveying and integrating contextual information from other brain areas. So this brings us to next consider what are the empirical evidence that we can in fact extract these spike information from layer 1 neurons, since they lie, layer 1, again, they lie conveniently quite close to the peer surface. So I will now move on to present a few findings, including our own, that spike activities likely from these layer 1 neurons are in fact present on micro-ECOG arrays. So in this study by Codagoli and colleagues, they have used in anesthetized animals micro-ECOG arrays to capture spike signals from the cortical surface. And their micro-ECOG system, which they term neurogrid, captured spike signals consistent with single unit activities in rats, as well as in human patients undergoing epilepsy surgery. So these spike signals could be spike sorted in pretty much the same way as traditional spikes recorded from intracortical arrays, as shown in this example here, on the bottom right image, that there are two distinct clusters of spikes with different spike symmetry and peak trough latency. So another example done at UCSD by Hermits and colleagues has performed simultaneous recording through intracortical probe and micro-ECOG in songbirds in an area called HVC in response to recorded audio of their own song. So the top spectrogram shows that of the actual bird song, and the signal traces in orange and blue are those recorded from the surface ECOG and the depth electrode respectively. And just by looking at the raw tracer, we can already see clear evidence of spike activities in many channels of the surface ECOG. And we can further see that spike signals from the micro-ECOG arrays, as per the image on the right, have very similar time course in milliseconds as signals from the deeper layers, but with different morphology from those neurons. And furthermore, there is actually little time lag between the signals recorded at the surface, the location S here, by the micro-ECOG array, as compared to those at the deeper area, D1, which is fairly consistent with the fact that these neurons are monosynaptically connected, as shown in many histological studies from the other groups that I've shown earlier. And moving on to some of our own work, the previous experiments were all done in anesthetized animals. So we wondered whether it's possible to also perform such recordings in awake behaving animals in a chronic setting. So to this end, we chronically implanted micro-ECOG arrays in the primary motor cortex of rats while they perform a number of behavioral tasks. So one of the tasks is the open field task, a simple task that does not involve any kind of pre-training and essentially only required the rat to perform spontaneous locomotion within a rectangular enclosure while recordings were done using the implanted micro-ECOG array. So movement was video recorded and the movement trajectory was processed with deep net cut and automated computer vision software algorithm. So this was the configuration of the micro-ECOG array that we used, consisting of 32 channels with each electrodes measuring 30 microns in diameter, so very small diameters, and spaced 400 microns apart. So this was roughly in line with the natural spacing between cortical motor columns as described elsewhere. The diagrams in the middle show some of the extracted spikes recorded from the micro-ECOG arrays during one of the behavior sessions in a single representative animal. So on the plot on the right, it shows that the aggregate signals across all sessions in 4 experimental animals. So these spikes have characteristics, namely the half-peak width and trough-to-peak latency, consistent with other published results. And the colors here each represents the sorted single spike units across different rats across their respective test sessions, noting the slight variation in spike profiles between the different neurons. Although broadly speaking, the spikes have a similar morphology and time cross across the animals. And the strongest evidence that these spikes are neural in origin and contain meaningful information is the fact that they correlate to different states that the rats were in. So if you remember the previous study where play cells-like activities in the monkey motor cortex were recorded as the monkey navigated a wheelchair to pick up grapes, we run a similar experiments in our rats, except that they freely move around and explore a rectangular enclosure while the micro-ECOG array reads out the layer 1 cortical spikes. And we designed a decoder that used these neural signals to predict the rat's position in the next 100 milliseconds. So here I'm showing a sample segment of spatial positions within the rectangular enclosure in A, and X and Y are the actual dimensions of the box. And the actual positions are plotted in black, and the machine learning predicted positions in red. We can also map out the X and Y positions separately as time series, as in figure B here. And you can see that the actual and the decoded positions mirror each other very well, with Pearson's R exceeding 95%. And figure C shows the same thing, but plotted as correlation plots. And again, lastly, we use a principal component analysis to show that there were clear differences in the ensemble spike pattern between the different spatial positions. So here we also look at the response properties of individual neurons. And each box here represents one sorted spike or potential single unit. And the color represents the spike rate, the firing rate of that isolated neuron when the rat was at different positions in the rectangular enclosure. And the layout of each heat map represents the actual spatial location in the test enclosure. So for example, the neuron on the top right has the highest activity when the rat is moving around the upper right corner of the enclosure. And overall, we can see that while there is an imbalance of tuning property in favor of the upper right corner of the test enclosure, and that the granularity of the place map is not the most precise, that's okay, as the neuronal population does have a range of location selectivity. And this ensemble of neurons contains enough information for us to decode body position in space quite accurately with the decoding. So there are a few caveats that we need to bear in mind with these micro-echo arrays. And one limitation is that the ability to detect spikes is predicated on the requirement that the electrode lies very close to the neurons in layer 1. So the array is therefore very sensitive to the presence of CSF, for example, between the electrode and the brain. So just a thin layer of CSF will shunt the electrical signals away from the electrodes and reducing the recording amplitude. So there may also be inflammatory reactions, for example, between the electrodes and the brain surface that increase the distance from the electrodes to the recorded neurons. Another theoretical limitation is despite the evidence that I've presented from other studies and our own, we still don't fully understand the whole range of response behaviors of these superficial layer 1 neurons, at least not to the same extent as the traditional layer 5 pyramidal cells, for example, or how reliable or robust they are when it comes to controlling BMI. So future studies should hopefully help to characterize this group of neurons better. So using thin film arrays will improve the conformity and the adherence to the brain surface by surface tension. You can see in this video that the thin film arrays adhere to the brain through surface tension and moves and pulsates with the brain pulsations. And in our rat experiments, in addition to having to using a thin film array, we put on a piece of gel foam, which is a kind of sponge, surgical sponge, which expands slightly to hold the array down quite firmly onto the PR surface. But we did notice that over time, we tend to lose spike signals, um, either from the dehiscence of the array from the brain or from inflammatory processes. So this is an area that needs to be further optimized. And on the image on the right, flexible arrays like this hug the brain contour quite well and can even be inserted into sulcus folds like this, as you can see on the bottom kind of left corner. Um, and lastly, while spike signals, specifically single unit spikes, are probably the most interesting signals, in my opinion, because it represents the activity patterns of a single neuron. We should also bear in mind that it is not the only type of neural signals that can be recorded from the surface. And one kind of signal is dendritic calcium spikes from deeper neurons, which happen over a longer time scale, over milliseconds. In this example, Suzuki and Larkin optogenetically excited layer 5 neurons and measured both LFPs at 600 micron depth, as well as with surface grid as shown here. You can see that the surface grid actually picked up dendritic calcium potentials that are time aligned with the LFP. And this dendritic calcium spike increases and decreases over a time course of 100 milliseconds, much longer than a single unit spike. And local administration of baclofen completely abolishes this calcium potential, as seen on the bar graph here in part C. They also elicited sensory evoked dendritic calcium spikes by applying an electrical current to the skin and measuring cortical responses, and again demonstrating that fairly large amplitude and slow spikes, calcium spikes, can be captured. So unlike single unit spikes, these are summated activities of collection of dendrites projecting in the vertical direction, along the vertical column. And another possible waveform is axonal potentials for axons that travel very close to the PO surface. Their activities can actually be picked up by the micro-echo arrays. And these are classically described as short duration triphasic waves due to the rapid polarity reversals at the nodes of Ranvier. So micro-echo array therefore has the potential to extend the spatial resolution to achieve essentially the same kind of spatial resolution as intracortical arrays, with the added advantage of being able to scale up to cover potentially a large area of the cortex, at which point the wiring and data transfer rate probably become more of a problem than the actual electrode size and dimensions. So granted that the response characteristics of layer 1 neurons are going to be different from that of the pyramidal neurons, we and the other groups have shown that rich information can still be decoded from these superficial neural spikes, and they in fact likely mirror the activities of pyramidal neurons in the same column. And in addition, we may even be able to decode non-conventional information due to long-range axonal connectivity from other brain areas that directly synapse onto these layer 1 neurons, as shown in many tracer and histological studies that I presented. So considering all these, it is therefore possible to design a high-density micro-ECOG array to span a large swath of cortex that may not be otherwise possible with penetrating arrays due to the cumulative risk of tissue injury. And study of single unit spikes over a wide area would potentially open new areas of research into different brain circuits. So here's actually my last slide, and here are some key take-home messages. We and other groups have demonstrated that spikes can be recorded from the cortical surface as long as the electrodes are small enough, appropriately spaced, and remain fairly close to the brain surface. And micro-ECOG arrays do not penetrate the brain parenchyma, so they are scalable with little incremental risk. And we should also consider the columnar organization of the cortex and that superficial neurons have similar tuning properties as deeper neurons, the latter of which are traditionally recorded by intracortical arrays. Layer 1 neurons convey information derived from other brain areas as well, which we know, but there is nonetheless a need for more studies into these areas in the future. And finally, micro-ECOG technology can potentially lead to new applications in BMI by enabling recording of neuron-level activities over a wide area of the brain that may not be feasible or safe to do so with penetrating arrays due to the need to disrupt the brain tissue and the cumulative risk of injury with that approach. And with this, I thank everyone for listening in, and I look forward to your questions and comments.
Check. Thank you very much. Fantastic work that you're doing here in Singapore. The institute looks also very nice. Alberto is asking, so if you can also click on the Q&A, that you can also see the questions yourself. So he's asking, so if deep neural tissue behavior can be implied by the behavior of coupled surface neurons, has there been any research on applying these techniques to detect and study DTIs?
By DTIs, do you mean diffusion tensor imaging, or is it something else?
I think so.
Because diffusion tensor imaging looks at like axonal bundles, the white matter tracks that go down, whereas for micro-ECOG arrays, they still record neuronal potentials generated from the cell bodies of layer 1 neurons. So they are kind of different modalities to begin with. But I think if I'm understanding your question correctly, there are potentially ways to correlate these signals in future studies. For example, in patients with chronically implanted ECOG arrays, and I guess we can also look at the DTI configurations. But having said that, I think DTI is a static imaging. It is based on structural considerations like the fractional anisotropy of water diffusion. So I'm not sure whether we can actually have any kind of functional correlation with that kind of approach. If that answers the question.
Thank you very much. Then somebody, I guess from Japan, is asking, how large can you make these arrays?
Yeah. So again, I'm a neurosurgeon, so I actually don't know the exact details of the engineering side. But I guess in theory, I don't think there is, in terms of the actual arrays themselves, I don't think there is an upper limit. You can have a very wide array that covers essentially the whole hemisphere, for example. But from what I understand, again, I'm not an engineer, the wiring bonding and you're gonna end up with thousands or tens of thousands of wires, which is gonna present a challenge in terms of how you wire that. I think that might be, and also the data transfer rate, when you're dealing with very high channel counts. But I think those problems can be solved. So that's why I think that getting single neural information over a wide brain region is the main advantage of such micro-electron arrays going into the future.
And what
And
is
I'm not sure about the cost as well. Yeah.
Yeah, do you have an idea about the costs of such an array?
I don't think I can give an intelligent reply to that. Sorry. Yeah, I don't know.
So a macro array or a clinical standard array with 64 channels is around $1,000, roughly. But the smaller arrays with more channels get much more expensive. So I know Graf Schalk paid more than $20,000 for a 256 channel array, but this was still not a micro one. But it's for research at the moment, so the costs come down if more people need it. Somebody's asking if it's equally invasive as normal ECOG.
Yes. They are there are surgical techniques to have minimally invasive options. Like there are like ECOG arrays which can be inserted through basically like a pinpoint, and they will inflate, they will they will unroll once you are inserted into the brain. But you can do that with conventional ECOG arrays also. So pretty much in terms of invasiveness, they are pretty comparable. And again, if we want to deploy over a large surface area, probably you need to do a large craniotomy. I mean, the risk of doing a craniotomy without disrupting the brain tissue is actually fairly acceptable, and neurosurgeons do that on a fairly routine basis.
I think the risk is comparable to anesthesia, so it's pretty low. Or you can also see differently. Anesthesia is pretty risky. But a big problem for standard ECOG grids is that you have to remove the dura mater, for example, from the bone, if you want to shift it between the dura mater and the scalp. And that's the reason why people do it under the dura mater, because then you don't have to rip up the dura mater. And for the micro ECOG grids, of course, the opening that you have to do is much smaller.
Yes, and also for micro ECOG, ECOG sometimes you can lay on the dura without opening the dura. But for micro ECOG, especially if you want to record spike information, it has to be directly on the brain surface. So you will never get any spike if this array is on dura.
Yeah, it's so small. Then Horst is asking, how does the array deal with different surfaces? The valleys and so on, so it's not smooth.
Yeah. So the answer to that is very flexible thin film arrays that contour to the brain surface. There may be biomaterials which will enhance the adhesion between the pia matter and the brain. So and also, as I've shown in the previous slide, I can go back. Like here, for example, these arrays can actually be folded up if they are soft enough and be tucked into the sulcus to record from these areas.
I guess if the electrode array is small, it's easier to insert it and to attach it to the cortex compared to the big ones.
Yes. Yeah.
So how soft are they?
They are super soft, and to be honest, I've accidentally torn some of them just by touching them. They are like I think on the order of like 10 micron or so. I'm not sure. Off the top of my mind, I don't think I remember the actual number, but they are micrometers in thickness.
And Said is asking exactly about this slide. Isn't the top of the electrode golden?
It can be gold with a P dot layer on top, but there are other designs, I believe.
Is it reacting with the body and the cortex?
With appropriate coating. So if we can show So one common way, which I understand, is that you can have polymer coating. Like for example, here you can have a gold electrode. I'm not sure whether you can see my cursor. I mean, you can have a gold electrode, but it's coated with P dot polymer. So this kind of prevents erosion of the metal and it improves the longevity of the implant. And again, there are other designs to encapsulate the electrodes.
Have you used any nanomaterials for the microarrays?
Not sure what you mean by nanomaterials, but like you mean like what, graphenes or something? No, I don't have any experience in that. But I think I heard of other groups using that kind of material, which I think is really promising.
So what are the most commonly used materials?
I believe is the standard gold and P dot with pyrrolene for insulation. That is the usual combination. Yeah,
What about platinum?
possibly. Yeah.
Rodrigo is asking, when it comes to clinical application, what type of application can you imagine in humans where microelectrode arrays are used?
Yeah. So anything in principle that the intracortical uter arrays can do. So control of like robotic neuroprosthesis or like screen cursors. Because like what I believe that layer one neurons share like great deal of similar response characteristics as deeper layer neurons, which are the targets for intracortical arrays. Again, like I mean, we still need to prove this because micro-ECOG is still relatively new. But based on what we know and based on our own findings, they have very similar tuning properties and firing properties.
Then Jan is asking, how does the signal quality change over time?
Yeah. So the signal quality, from our experience, you tend to lose. I mean, we tend to lose the spike signals after I guess, a few weeks. Which again, as I said, we need to optimize that. But I think the reason for signal loss may be different from intracortical arrays, because I feel that over the long term, these arrays, because of the materials that we currently use, they tend to dehisce, they separate from the brain, therefore losing the spike signals. Although the local field potentials can still be captured. Whereas for intracortical arrays, because you are disrupting brain tissue, inflammatory reaction is stronger. Even though possibly micro-ECOG arrays may also generate a little bit of inflammatory reactions, but I don't know for sure. I haven't really looked into that yet. But it's for sure is less than the intracortical penetrating arrays.
This relates to the next one. So are these arrays moving?
They shouldn't move, to be honest. They should be stuck to the brain surface.
And then Jakob is asking, can they be implanted on the cerebellum?
I believe yes, because on the cerebellum you also have the layer the cortical the layers are very different from the cortical layers, but you also have very superficial neurons, which activities you can measure with these arrays. But that will be a quite interesting kind of experiments to perform.
And somebody's asking, what was pulsating in your talk?
Well, is for that short period, that rat was breathing, so the pulsation was breathing artifacts. So the brain some kind of pulsates when the rat was breathing for that instant. Yeah, it doesn't always happen, and we don't see it that obvious in most cases.
Which artifacts do you see in your data?
What artifacts?
On surface recordings, you get movement artifacts and
Yes.
noise artifacts. So which ones do you see?
For sure, movement artifacts. So we have to actually eliminate those recording epochs with large movement artifacts. Um, Again, I feel that because we are using a wired setup, a lot of these movement artifacts are slightly from the cable. So converting to a wireless setup may help to reduce some of them. And of course, we have the usual the environmental electromagnetic kind of artifacts, and physiological artifacts like cardiac. But all these can be actually subtracted out, because if you're only looking at spike signals, you can do kind of like bipolar referencing or other techniques to reduce these artifacts, including movement artifacts, if they affect um, the channels to similar extents.
Then Martin is asking, how widely used is this technology currently?
In clinical practice, not that I'm aware of. I don't think there is. I may be wrong. I mean, I'm not aware of any human patients, BMI or BCI applications with micro-electrocardiogram arrays. Yeah.
And how many research groups are working on that?
For prostheses in Singapore.
On the micro-electrocorticographic.
You mean around the world? I
Yeah.
think there are at least like what I presented. I guess 4 or 5 main groups. Yeah.
Did you control prosthetic devices?
Can I control? I don't have personal experience, but it's an area that I'm looking into. And in Singapore, there are other groups that work on neuroprosthesis. Yeah, and what is efficient? Well, like how the signals compare to spikes, to the intracortical spikes need to be further tested, um, because while there are response similarities between the deeper neurons, there are also differences. And also layer one neurons have a wider kind of like connectivity from different regions. So you may get a lot of like representational interference from other areas. The decoding can be more difficult, and more non-linear, for example. There are some considerations for that. So again, these are all ideas for future research.
Then somebody's asking, do you think micro-electrocorticographic would be a good choice for studying self-induced afferents copies?
I'm not sure I'm not sure if I understand what you mean by that. Self-induced afferent copies. Could you elaborate?
Yeah.
Yeah.
Let us know what you want to know. So did you see we had more than 70,000 people watching Spring School already?
Yeah.
So it's a lot,
It's a
especially
lot. Yeah.
in Southeast Asia. A lot of people are joining in.
Yeah.
So your talk will be seen by many, many people.
Great. Thank you.
So it was very nice to have you, to see you again. Pleasure.
Yeah. It's a pleasure to
Fantastic
be here.
talk. Look at all the chat comments. Thank you for this amazing work and so on. And yeah, they are visiting you in Singapore to talk in person. This is even better. So it was a pleasure to have you. Thank you very much.
Thank you very much.
And enjoy the day. And we are moving all the way from Singapore to England. Raini, can you already hear us?
Yes, I hope you can hear me as well. Hey, Christoph.
I can also
hey,
see
audience.
Hey, you are talking today about micro recordings with spikes.
Yeah.
maybe you should share your own slides. Makes it easier. Nice to see you.
Share the screen. So someone is still, I stopped the screen sharing. Whoever's sharing the screen, I stop you. What's my screen? This one here, share.
Please share also audio if you want to have video sound. So Raini is actually from South Tyrol in Italy, and then he moved to University of Technology in Graz. That's the reason also why I know him. And a couple of years ago, he moved to Essex, and now he's working and leading many projects in, I think it's the biggest BCI group in England, isn't it?
Yes.
Yeah. How many people do you have now? It's like 50 or it's a lot, right?
I mean, our email distribution list has at the moment 43 people, but there are some visiting and PhD students who are not in there. So it's